So, the typical explanation of how a cell gets power in your biology textbook is something along the lines of there's a membrane, there's electrons on one side of the membrane, there's no electrons on the other side of the membrane, and there's a series of channels that the body uses to bring those electrons into the cell to power all of the whatever that all of the stuff. There's a ton of stuff that has to go on inside each individual cell, and that's what ATP does. Is it donates the electrons basically for the electrical charge. So what you're saying is, or for I guess I should say for the chemical charge, just to keep it separate. But what you're saying is that those channels, those things that are actually bringing in the electrons. Those aren't like a biological physical structure. That this is the whole process is being mediated by these lattices of fourth phase water. That's what's allowing these electrons to pass through the membrane. Am I close?
Uh,
The look on his face says no, guys. I don't know if you're watching the video. I wasn't even close.
Okay. I mean, there's a lot to say about it. The first question is about the channels.
Okay.
And I'm sorry to be so radical in my thinking, but
no, you're fine. I'm trying to get to the bottom of this.
Well, I think the channels don't exist.
Okay. Oh, okay. That is radical.
Yeah, I mean now I've lost total count, but I think the number of channels that are proposed to be in the membrane somehow exceeds 1,000 or something more than that. And the first question is there enough room for all of these channels in the membrane? Okay, this is a serious consideration. But more than that, think of the logic. So each channel is specific for a given solute or a given ion. Right? Otherwise, you would need a separate one for each ion. And think of the problem. Think of the channel that's specific for the largest solute for which there is a putative channel. Okay, so let's say it's the size of a basketball. So you've got a channel and you've got basketballs outside the cell, and the channel pops open and the basketball goes through. So when it opens to let the basketball through, the question is, how does it prevent the golf balls and softballs and hard balls and such from passing through at the same time? So you have to invent a mechanism that's capable of doing that. And it's not just capable of doing it for the basketball, but for the next smallest ball, you got to do the same thing for all the solutes that are smaller, and the next smaller one, et cetera, et cetera, et cetera. So it means the burden on figuring out a mechanism as to how this could possibly work, it boggles the mind. And I do know that there's one proposal about sodium and potassium, distinguishing those two, but it's not just for those two, it's for every single channel that necessarily must exclude all the solutes that are smaller. It's kind of like the dog door that you have, in your house, so the dog can go out or in. And when the dog door opens, what prevents the cats and the mice and whatever from going in and out at the same time? It's that kind of problem. So it's illogical. And let me say one more thing. The membrane is said to have pumps and channels. So the pumps are for pumping various ions and solutes in or out. And I'm told by students of this that there are now more than something like 2,000 of these. Imagine 2,000 of these spread around the surface of a cell. And Gilbert Ling himself, in his own experiments, he pulled the plug on all the power to the cell. In other words, he poisoned the cell. And though he poisoned the cell, still the apparent effects of pumping were still felt for 8 hours or something like that, the separation of ions. And he said, well, there's no energy to run these pumps. Pumps need energy. And so that was when there was only one pump that was identified. It was the sodium pump, and now we have 2,000 of them. So if there's not enough energy to run the sodium pump in the cell, how are you going to run 2,000 pumps? So the whole concept, to summarize, the whole concept of channels and pumps, although widely accepted by almost everybody, I think is erroneous. And also, just I guess a final point on that is, you can compare a cell to a gel. The inside of the cell has a membrane, and all of these various pumps and channels are said to lodge in the membrane, and they're responsible in theory for the negative electrical potential inside the cell. But if you take a gel, has no membrane at all, you find the same electrical potential, more or less, inside the gel as inside the cell. Many people, including our lab, have measured this. So if the cell is, say, minus 80 or 90 millivolts, the gel is minus 100 or 110 or 130, has no membrane. So if anybody is suggesting that in the cell, it's the pumps and the channels that are responsible for creating this electrical potential, then there has to be another mechanism for why the gel has the same electrical potential, although it has no channels and pumps, obviously, because it has no membrane. So I'm pretty skeptical about the current view on why the cells all have negative electrical potentials. And I think a simpler argument stems from the idea that the water inside the cell, practically all the water, is easy water, which typically has negative charge. So it's a very simple kind of interpretation. And if your cell has less negative charge, smaller negative electrical potential, then it has less easy water.